we'll finish the metabolism today probably. You know, a lot of these things you already know. So it's kind of more of a refresher. So we stopped the previous episode of the story about metabolism at the moment when glycolysis is over, Krebs cycle is over. We've got a whole bunch of electron carriers, NADH and FADH2, that hold the electrons to different places in different cells. In eukaryotic cells, what those places are going to be? Where the electron carriers, where do they go? The matrix of mitochondria, right? Krebs cycle in eukaryotes is already in mitochondria. Okay, so all those electron carriers are channeling towards the inner membrane of the mitochondria. What about prokaryotes? Do they have mitochondria? Do bacteria have mitochondria? No, they don't. Do they have membrane? Oh yeah, they do. They have cytoplasmic membrane. That's where these processes occur. So actually, the final stage of the oxidative respiration, aerobic respiration, happens in the membrane. Okay? So what essentially is going on, the NADH and FADH2, when they deliver the electrons to the assembly of proteins called electron transport chain, NADH and FADH2 are essentially oxidized because they lose electrons. That makes sense? What is reduced then? Where do these electrons go? To the proteins of... Yes, proteins of electron transport chain. Now, if you would look at the events that go on in the electron transport chain, essentially, you can trace it. That's a little arrow that shows here. So, electron goes from one protein to another. Essentially, one protein is oxidized by another, okay? And as electron moves through the proteins of electron transport chain, it releases its energy. Does that make sense? Something like water that falls in the wheel of the water mill will release its energy and will rotate the wheel. Does that make sense? You got it? The whole idea of electron moving through the electron transport chain. Question? Yes. Sure. Absolutely. Absolutely. Now look. We've got NADH and FADH2. Is that reduced or oxidized form? Say again? Reduced, because they carry electrons. The purpose of those molecules is to carry electrons to electron transport chain. So the electron from NADH goes to the, I don't remember the names of those factors, they're like 1 kilometer long, and of course I'm not asking you to know them. Anyway, so that is the electron from NADH. And green is going to be an electron from FADH2. Okay? And those electrons, they move via the proteins of electron transport chain from one to another. Does that make more sense now? Or not? Okay? They kind of like jumping from one step to another of a stairwell. And with each step, they release energy. This energy is used by the proteins of electron transport chain to pump hydrogen ions across the membrane, essentially concentrating hydrogen on one side of the membrane and depleting the hydrogen concentration, depleting hydrogen ions on another side of the membrane. Does that make sense? When you have high concentration of hydrogen on one side and lower concentration of hydrogen on another, what do you create essentially? A G-word. Gradient. You create a gradient. You agree with me? Gradient is chemical, first of all. Chemical because hydrogen ion is the chemical. And it's also electrical because you have more positive charges on one side than on another. It's called electrochemical gradient. Each gradient, what does it sort of want to? You have a gradient, high, low. What's going to happen? Yeah, it wants to equalize, okay? It wants to balance it. So hydrogen well, they don't want anything, but it has a tendency to move from high to low concentration. Does that make sense? Can hydrogen, positively charged ion, move directly across the membrane? Positively charged ion. Can it move directly across the membrane, hydrophobic membrane? No, it cannot, because it's hydrophobic. It doesn't let any charged particles to diffuse directly. If it cannot diffuse directly, what is it going to use to diffuse through? Transmembrane protein. Yes, transmembrane protein. Thank you. So this transmembrane protein is called ATP synthase. I really like how it is represented here in the form of a distorted pair. It's an enormously complex protein. It contains a part through which hydrogen ions diffuse. And as they diffuse, bless you, essentially going down the slide, during the diffusion, they release energy. Does that make sense? It's pretty much a kinetic energy of ionic movement. That energy is then utilized by a synthesis part of the protein and is used to produce ATP. Energy that protons release when they diffuse through the ATP synthase is used for essentially the ATP synthesis. Does that make sense to you? No? Okay, think about this. I'm not gonna go ahead. No, I'm not gonna draw this stuff, but it's kind of, imagine that I am the electron transport chain, okay? And I have my lunch. Lunch gives me energy, okay? And I have a pump. So I pump protons high. Okay, I use energy that was in the lunch to pump the protons. Is that clear? Electron transport chain uses the energy of electrons to pump protons. Protons then start to fall down through the ATP synthase. And they fall down and they roll the wheel of the watermill, which is producing ATP. So that watermill, the wheel, converts the energy of falling protons, the kinetic energy of protons, into the energy of chemical bonds in an ATP molecule. Does that make sense now? So the direct source of energy for ATP synthesis is the proton diffusion across the membrane. But this diffusion is possible because we have electron transport chain that creates the gradient. And it's a never-ending process. Does that make sense? Okay? Understand this idea here. What happens to NAD plus and FAD, by the way? after they delivered products? What happens to them? Any idea? It's not in the slide. You go back to hold another load of electrons. Kind of shuffle. They like taxi. They shuffle back and forth between the Krebs cycle and electron transport chain, back and forth. Does that make sense? Okay, good. So that's a summary. Okay. Essentially, if you would look at the entire process in the sense of what is going on, what is the bigger picture of the aerobic respiration, you're going to see that glycolysis is prepare pyruvates, get some NADH. Transition, convert pyruvates in the form that is suitable for Krebs cycle, churn out some NADH. Krebs cycle, finally complete oxidation of glucose, churn out NADH and FADH2. And in the brain freeze, oxidative phosphorylation, electron transport chain, these guys, okay, they will yield the major amount of ATP. That's when all the ATP is synthesized. Does that make sense? Now, where does electron go? Where does electron go? Electrons are bad. I mean, they're not bad, but good God, they're active. Where does it go? The electrons from electron transport chain. They just hang out in the cell, crushing everything on their way. They have to stick to some molecule. Which molecule do they stick to in aerobic respiration? In aerobic respiration, oxygen. They reduce oxygen. Does that make sense? Okay? They reduce oxygen. Electrons, how are they charged? Negative. So if they reduce oxygen, they give it also negative charge. Do we have to compensate it? Well, preferably, yes. Which abundant particle carries positive charge in the cell? Ion. Ion. Hydrogen ions. Hydrogen ions, they go back into the cell, and when you have hydrogen ions, oxygen, and electron, you end up with water. Does that make sense? So, oxygen in oxidative respiration is called final electron acceptor. You know why it is called final electron acceptor? Because he's the, well, this molecule is the hungriest for electrons. Doesn't make sense what I just said? It's oversimplification. Each chemical that can oxidize can be characterized by the parameter called electronegativity. Electronegativity means how strong the attraction of that, how strong this molecule attracts electrons. Okay? Oxygen is extremely electronegative. It takes electrons from everything, it oxidizes everything. Does that make sense? Oxygen is extremely oxidative. Okay? It's much more oxidative than anything that accepts electrons in a cell. So it is the final acceptor. It finally gets all the electrons to it. Does that make sense? Strongest electron acceptor. And I want you to keep this in mind, that oxygen is the final electron acceptor because it strongly attracts electrons. Okay? It's not electrostatic, it's not like positive, negative, it's different mechanisms, quantum mechanisms. Now, what other strategies for ATP production can cells employ? There are generally three types. So we talked about aerobic respiration, right? With three stages: the glycolysis, the transition state and Krebs cycle, and then electron transport chain. Some cells use anaerobic respiration. Okay, so let me just fill it out. So in aerobic respiration, final electron acceptor is oxygen. Say it out loud. In anaerobic, when we say anaerobic, what does that mean? Without oxygen. So it should be something else. And that something else is fairly oxidative, inorganic molecule. For instance, sulfate or nitrate. They're still pretty damn oxidative, just not as good as oxygen. Does that make sense? So if you would look at the process of anaerobic respiration, conceptually, it consists of all the same parts: the glycolysis, the Krebs cycle, the electron transport chain. But the difference is that the final electron acceptor is not oxygen, some other inorganic molecule, which makes anaerobic respiration less effective in terms of ATP production than aerobic one. Now, tricky question. No, not tricky, but complex question. Kind of, you have to do the leap of logic. Why anaerobic respiration doesn't produce quite as much ATP as aerobic does? Hint, think about electron acceptors, final electron acceptors. What is the final electron acceptor in aerobic respiration? Oxygen. In anaerobic, say, pick one, nitrate. Which one has more hunger for electrons? Oxygen. So oxygen can, quote unquote, extract more energy from electrons because it's hungrier. Does that make sense? Anaerobic respiration may come close, but will never match the efficiency of aerobic one. Does that make sense? In fermentation, the final electron acceptor is some sort of organic molecule. For instance, acetaldehyde. In alcoholic, in general, in fermentation, what you end up with, you do the glycolysis, and that's pretty much it. Maybe a couple more reactions after it. In fermentation, there's no Krebs cycle. There is no electron transport chain. So in terms of efficiency, in terms of number of ATP molecules produced in glycolysis, how many do we have in glycolysis? How many ATPs are produced during glycolysis? 2. It's really little compared to about 38 molecules produced in the process of aerobic respiration. What is the advantage? How fermenting organisms compensate for a low yield? Oh yeah, it's much faster. You do glycolysis, 2 molecules. Glycolysis, 2 molecules. Run, 2 molecules. Here in aerobic respiration, you got to go through glycolysis, Krebs, electron transport chain, chemiosmosis, the diffusion of protons through the membrane, all those steps to get a lot. So it's slower. Does that make sense? Now, is there a difference between aerobic and anaerobic respiration other than fine electron acceptor? Yes, ETC, electron transport chain, should be different. Because they channel electrons to a different molecule, the enzymes are different. This pumps, the proton pumps are different. Why there are anaerobes? What's the reason organisms cannot use oxygen? First, there's one very, very simple reason. Yeah, because there's no oxygen in the environment, okay? If, I don't know, if you live in the human gut, it's extremely anaerobic condition, okay? Some microbes do not have genes that encode electron transport chain, okay? Like cytochrome oxidase, the last protein that transfers electrons to the oxygen. What is also important, during the aerobic respiration, cells, bacterial cells, produce huge number of reactive oxygen species. One such reactive oxygen species should be in your medical cabinet at home. It's hydrogen peroxide. What do you use hydrogen peroxide for? You can do it for cleaning to kill what? Bacteria, right? So you use hydrogen peroxide to kill bacteria, and it turns out bacteria do produce it. So they have to have a system to deal with it. Correct? They have to have a system to neutralize hydrogen peroxide. And there are enzymes, catalase and peroxidase. Well, I don't have catalase here. We're going to talk about it in the next chapter. But catalase and peroxidase, they deal with hydrogen peroxide. Superoxide dismutase deals with a superoxide anion, which is crazy dangerous. Okay, it can oxidize the hell out of everything. Okay, so if microbe has genes for these proteins, it can be aerobic. If it loses genes for these proteins, it cannot be aerobic, it cannot do the respiration. Microbes that lack all three genes will be obligate anaerobes. They will get killed by oxygen. Okay? We'll chat about it when we'll talk about growth conditions. But think about this. It's all about phenotype. It's all about having a certain gene for a certain enzyme. Whether it's an enzyme of electron transport chain or the enzyme that neutralizes reactive oxygen species and whatnot. Does that make sense? Fermentation. Just a few more words. We're probably not going to finish it today, so relax. We'll finish it tomorrow. No worries. Fermentation. Glucose is getting oxidized, but this oxidation is incomplete. I want to mention two major types. Fermentation, the lactic or acidic fermentation, should I say. But here we talk about lactic fermentation and alcoholic fermentation. Okay, lactic fermentation gives us a lot of wonderful products, like buttermilk, okay, yogurt, cheese, sauerkraut. Alcoholic fermentation gives us alcohol. And I know that there are cases discovered and described in literature of people who somehow acquired microbes capable of alcoholic fermentation, and those microbes established themselves in the guts. And it kind of sucks. Well, from one, somewhat, because you constantly buzzed. You have yeast that produce alcohol, and you constantly have alcohol coming into your blood, in your intestines. It's not that you're like totally drunk, like wasted all the time, but you have elevated alcohol concentration in the blood, and it may actually cause problems, when you're driving. Okay. Now this is an example of classic alcoholic fermentation. It's lactic fermentation, but that's not it. That's not all the products that we get from fermentation. Nail polish, the acetone, is produced in one kind of, I think it's some sort of a Clostridium. Yeah, I think it's Clostridium acetobutylicum that produces acetone. Okay. Some very specific microbes are required to produce Chardonnay because they produce certain species of certain chemical compounds that give Chardonnay wine a taste. Clostridium butyricum participates in the fermentation of milk that results in butter. Vinegar, produced by microbes. Swiss cheese, oh, that's my favorite. Propionibacterium gives the Swiss cheese a very particular taste and smell. Not everybody likes it, to be honest. I have a very good friend. He's not Swiss, he's Georgian, not from Atlanta, Georgia, the country in Europe. Okay. His mother once brought the national, local goat cheese. My wife, quite frankly, said that it smelled like 2 weeks old socks. It tasted awesome, but sometimes the cheese smells as a very strong odor and very specific. So remember that mention about socks. Okay? Turns out that the genus Propionibacterium contains numerous species. Some of them are responsible for that taste and the smell of the cheese. Propionibacterium acnes is responsible for the condition that is called acne. It normally lives in the hair follicles. And when we go into puberty, we start to produce a lot of sebum, that oily substance that lubricates the hair. Propionibacterium acnes uses it as a food source. And when there's a lot of sebum, there's a lot of Propionibacterium. When there's a lot, really a lot of Propionibacterium, your immune system starts to recognize it. You have inflammation and you have a zit. If you pop the zit, rub it a little, whatever comes out of it. Zit, I'm not talking about carbuncle, I'm talking about the zit. Rub it, smell it, smells like a Swiss cheese. No, seriously, it smells like Swiss cheese. Now, regarding the fermentation, remember, the final electron acceptor is organic molecule, okay? In alcoholic fermentation, it's going to be acetaldehyde. In lactic, it's actually going to be pyruvate. An interesting fact that some microbes can use both fermentation and cellular respiration in different conditions. So think about this. You have a microbe that can ferment and can do oxidative respiration. When is it going to utilize each of these strategies? Yep. If microbe has oxygen, it's going to use aerobic respiration, right? And we'll get the energy from this. If there's no oxygen, it's going to use fermentation. Does that make sense? Okay. We're going to talk about this and wrap it up. I will not give you the quiz. I'll give it to you tomorrow. Okay, and you may say, holy crap, we have an exam on Monday. How are we supposed to know? Well, guess what? We're going to go over it on Monday morning. Okay? Does that make sense? Good. So, test is on Monday, Tomorrow is Thursday. Okay. I'm giving it to you tomorrow, and we will not see each other till Monday. But following our famous tradition to go over the quiz, we can go over it on Monday. Like in the morning, we get together, we go over the quiz, you give me everything, and we start the exam. Does that make sense? Okay, let's keep up with the proteins and lipids. Can microbes use fat as the source of nutrients, source of energy? Proteins? Of course, of course. Last semester we tested microbes that grew up on like 2 weeks old pork that was forgotten in the fridge. Pretty bad. And it smelled, oh my god, it smelled. So, how they metabolize. It will remind you, metabolic pathways are the same that are in humans. So lipids, triglycerides, okay, going to be broken down into the glycerol and free fatty acids. Glycerol will be, we don't have glycerol on this scheme, but glycerol will be converted into the glyceraldehyde 3-phosphate, which is the glycolysis intermediate. Does that make sense? I just don't want to go all the way back to glycolysis, but trust me, there is a glyceraldehyde 3-phosphate intermediate in the middle of glycolysis. Glycerol is converted to it, gets into the glycolysis, and then just supplies, additional supply for glycolysis. Got it? Free fatty acids are oxidized in the process of beta oxidation. Beta oxidation results in quite a huge number of acetyl-CoA molecules that can enter the Krebs cycle. Does that make sense? And when this acetyl-CoA is further oxidized in the Krebs cycle, you get a load of high energy electrons. Does that make sense? So know what triglycerides are metabolized into, glycerol and free fatty acids, and where do glycerol and free fatty acids go. Does that make sense? Proteins. Proteins consist of amino acids. Break down proteins into amino acids. What you can use amino acids for? Can you use them back for protein synthesis? Absolutely. But if you are short on nutrients, can you break them down? Sure, and you can break them down in the process of oxidative deamination. Okay? Deamination may either give you eventually acetyl-CoA, which enters the Krebs cycle, or in some shorter processes, it may generate pyruvate or amino acids like oxaloacetic or alpha-ketoglutarate that can, so not amino acids, but compounds like oxaloacetic acid or alpha-ketoglutarate acid that can get into the Krebs cycle because they are part of the Krebs cycle. Does that make sense? So products of lipid metabolism, glycerol after the conversions ends up in a glycolysis. Free fatty acids after the conversions end up essentially in the Krebs cycle. Products of amino acid metabolism can become pyruvate or can enter the Krebs cycle. Does that make sense? Okay? Questions about this? About this? I'm not going to ask very complicated chemistry. Okay. This is it.